purpose of the center is really to develop new approaches for treating individuals who are exposed to very high levels of neurotoxic chemicals that can trigger seizures. It's important for a number of reasons. First of all, there's a number of classes of chemicals that the Department of Defense has determined may be credible terrorist threat agents. In other words, these may be chemicals that groups who are engaged in terrorist acts against the public may use to intimidate the public and or cause increased deaths in the public. And so that is one major aspect of this project, is to really develop the medical tools that are needed not only by the doctors in the hospitals who are treating people who may be exposed to these chemicals, but also by first responders, emergency responders who are first on the scene. So having better medical approaches for dealing with these will not only protect these health people who are involved in responding to these emergencies, but also hopefully increase the number of lives that are saved and also improve the lives of those who survive exposure to these particular chemicals. There are two broad classes that we'll be investigating, and these are the two broad classes that are of most concern at the moment to not only the NIH, who is funding this project, but also to the Department of Defense. One group of these are known as organophosphates, and these are chemicals that inhibit an enzyme in the brain known as acetylcholinesterase. OPs were developed for use as pesticides. They've also been used as nerve agents during World War II. So these are well-known chemicals that have been around for a long time. There is a standard antidote out there which is issued, for example, to people in the military when they go into an arena where they expect they might be exposed to these organophosphate nerve agents. But there are a large number of people, a much larger number of people in the world who are exposed to OP pesticides. And there are a large number of deaths in the order of thousands that occur every year as a result of overexposure to these particular pesticides. The tools that we currently have to treat these people can keep them alive if they're issued soon enough after the exposure, but even at that point, if the person is kept alive, if they go into a serious seizure, there's often brain damage that results afterwards. So one of the goals of this center is really to develop better tools or to identify drugs that can be used together with the existing antidotes that are available to, again, improve the number of people who survive, and increase the number of people who survive, and also improve the outcome for those that do survive an exposure. The second class of neurotoxic chemicals that are of great concern to both the NIH and the Department of Defense are GABA receptor antagonists. Basically, these are neurotoxic chemicals that target a particular receptor in the brain, and you may know them better as rodenticides. So there's a number of rodenticides that kill rats or mice that work via this mechanism. The one that we'll be studying in great detail in our center is known as TETS. That's sort of the abbreviation for it. It is banned worldwide, but is still still available illegally in the black market. And so and it's easy to synthesize, so there's great concern that a terrorist group could gain access to this. Interestingly, there's still a number of deaths worldwide from this particular compound as well, because it is available illegally. And this has occurred primarily in China, where this particular drug is very well known, or toxin is very well known, but there are also accounts of TETS intoxications occurring here in the United States as well. These two groups of chemicals, the organophosphates and the GABA receptor antagonists, work through different mechanisms, but the end result is basically the same, which is to change the way the neurons in the brain, or the cells in the brain, talk to each other, and they increase the activity of this communication. So the neurons are firing more actively. This increases excitation in the brain. And if that level exceeds a certain set point, that can actually be sufficient to trigger a seizure. One of the other important things that this center is going to be addressing is not only can we identify drugs that will either stop seizures before they start following exposure to these neurotoxicants, but also can we find drugs that will stop seizures once they start, and third, can we find drugs that will stop the downstream neuropathology that can result following a very severe seizure. We have some evidence in our lab that these chemicals will cause neuroinflammation in the brain, and this may actually lead to some of the long-term deficits that we see in people that are exposed to these chemicals at high levels but survive. An example are the victims from the sarin attack in the Tokyo subway. Those individuals that survived that but did suffer seizures as a result have long-term problems with learning and memory. They have long-term problems with anxiety and with depression.